Good morning, viewer. Welcome to our FTV English version. Here's amazing news for our viewer. Yeah, erythritol. Erythritol may impure cellular functions essential to maintaining brain blood vessel health, according to researchers at the University of Colorado Boulder. Findings suggest that erythritol increases oxidative stress, disrupts nitric oxide signaling, raises vasoconstrictive peptide productions, and diminishes clot dissolving capacity in human brain microvascular endothelial cells. Erythritol has become a fixture in the ingredient list of protein bars, low-calorie beverages, and diabetic-friendly baked goods too. Its appeal lies in its sweetness to calorie ratio, roughly 60 to 80% as sweet as sucrose with a tiny fraction of the energy yield, and its negligible effects on blood glucose too. Erythritol is also synthesized endogenously from glucose and fructose via pentose phosphate pathway, leaving baseline levels subject to both dietary and metabolic influences. Concerns about erythritol's safety have escalated following epidemiological studies linking higher plasma concentration with increased cardiovascular and cerebrovascular events too. Positive associations between circulating erythritol and incidents of heart attack and stroke have been observed in US and European cohorts. Independent of known cardiometabolic risks, too, factors a causal mechanism for the link has remained elusive. In the study, the non-nutritive sweetener erythritol adversely affects the brain microvascular endothelial cell functions, published in the Journal of Applied Physiology. Researchers designed in vitro experiments to test the cellular consequences of erythritol exposure on cerebral endothelial function. Human cerebral microvascular endothelial cells were cultured and exposed to an amount of erythritol equivalent to consuming a typical beverage. Also, experimental conditions include 5 biological replicates per group. Cellular assays measures oxidative stress, antioxidant proteins expression, nitric oxide bioavailability, endothelin productions, and fibrolytic capacity too. Capillary electrophoresis, immunoassay, and ELISA were used to quantify expression of superoxide dismutase catalase too. For the same endothelial nitric oxide synthesis, phosphorylated ENOS, endothelin 1, and tissue type plasminogen activator also. Viewers, cells exposed to erythritol exhibited a substantial increase in oxidative stress. Reactive oxygen species levels rose by approximately 75% relative to untreated controls. So, antioxidant defense markers were also elevated, with SOD1 expressions increasing by approximately 45% and catalyzed by approximately 25%. BNS exclusive report, FTV English, Delhi, Bharat.